Hello, everyone. My name is Kerrigan Nelson, and I'm 21 years old. I am treated at Dana-Farber, and my oncologist is Dr. Janeway. Super lucky. I also sang last night, and I would rather, yeah. Thank you. When it comes to public speaking, I get really nervous. I would rather just sing everything. But right? Don't tempt me. I have been fighting osteosarcoma for the past 3 years. I've had amputation and lung surgeries, etc., all of that fun stuff. All right, and now, time for your introduction. All right. Dr. Isikoff is the division head for the Center for Cancer and Blood Disorders, the director in sarcoma program, and lead physician for the Reed R. Is it Sacco adolescent and young adult cancer program. Dr. Isikoff has a research interest in clinical trial development in sarcomas and other solid tumors and is the site principal investigator for the Children's Oncology Group in the Beat Childhood Cancer Consortium. In addition, he serves as chair of the protocol committee and the lead for the clinical trial development in sarcomas for the National Pediatric Cancer Foundation's Sunshine Project. So everyone give a hand for Dr. Isikoff.
I guess I'm using this. I'm not sure. That was like the best intro ever. And if you were there last night, you know that she is like the best voice ever. So it's my pleasure to be here. I'm very excited to be leading or moderating this session. I think, as a pediatric oncologist, for me, I get most excited about clinical trials and, bringing the therapies to the patients. But I recognize greatly that all of the talks leading up to this session really are so critical because we can't develop clinical trials without all that amazing science that we've seen. But I don't do any of that science. I like getting that information and then developing a clinical trial. And the folks at this panel are incredible at that also, and we're lucky to have them all here today. We do have a large panel. There's 7. So this session, you may notice, has 4 talks, and then there's a break, and then 3 more. So if you have questions during the first 4 talks, please write them down so that you can ask each of the doctors that are going to be presenting. So I'm going to go through real briefly and introduce everybody. Dr. Vishu Avutu is attending in the Department of Medicine and Pediatrics and is the founder or a co-founder of the Adolescent and Young Adult Oncology Program at Memorial Sloan Kettering Cancer Center. Today he'll be describing a phase 1/2 study of ZN-c3 plus gemcitabine in relapsed and refractory osteosarcoma. Next to Dr. Avutu is Lara Davis. She's an associate professor at Oregon Health and Science University, where she is the sarcoma program director and co-leads the translational oncology program at the Knight Cancer Institute. Most famous for Nike shoes, I'm assuming. Today she'll be reviewing the SARC-38 clinical trial, which is a phase 2 study of regorafenib plus nivolumab in patients with refractory or recurrent osteosarcoma. Dr. Nicola Mason is a professor of veterinary medicine at the University of Pennsylvania, and her research focuses on developing safe and effective immunotherapies for dogs with cancer. And today, Dr. Mason will be describing a multi-institutional clinical trial in pet dogs with spontaneous osteosarcoma that are treated with a recombinant HER2 targeting listeria. Virtually, actually our panel is even bigger than what you're seeing here. Virtually, we will have Dr. Rosie Kaplan, who's a physician scientist at the NIH, focusing on developing novel biomarkers and targets of the metastatic microenvironment in osteosarcoma, and she'll be telling us about next generation cell therapy for osteosarcoma. Dr. Katie Albert will be next, and she's an assistant professor and pediatric oncologist at Seattle Children's. She's the director of the solid tumor program and focuses on adoptive cellular therapy for high-risk sarcomas. And today she'll be reviewing solid tumor CAR T therapy from Seattle Children's Therapeutics. Next will be Bhuvana Sethi, and she is an associate professor at The Ohio State University. That's trademarked. If you use the word "the" in any sentence with her, you have to pay money, apparently, to somebody. But she's the director. I went to the University of Michigan, so I'm actually hating what I just said. But she told me I had to say it. So she's the director of the sarcoma program at Nationwide Children's. And today, Bhuvana will be describing a clinical trial aimed at harnessing natural killer cells for the treatment of osteosarcoma. And finally, Dr. Janeway in the end. So she already had an introduction. I was kind of thinking I should just think of something to say about Dr. Janeway that no one else would know, or I don't know. Dr. Janeway and I trained together at Dana-Farber, so I have known her for a long time. She was always smarter than me. She's nicer than me. I was going to go through, I have 17 seconds. Let's see. One time, no. Kitty owns like 16 cats. I'm not sure what that's about. That's not true. But I'm thrilled to have to be able to introduce Katie, who I, although trained together, she's one of the people I most respect, and am honored to be able to introduce her today. And she's going to be talking about the Children's Oncology Group trials and some of the work that's being done through the Children's Oncology Group today. So, Dr.
Good afternoon, everyone, and thank you for this opportunity, especially to the Make It Better community, and also Gavin's family for giving me this opportunity to come out here and talk to you guys. So I'm going to be talking to you about this clinical trial, and if any of you have any cool names, please let us know. We are actively looking. So first of all, I have no conflicts of interest to disclose, but I do want to acknowledge that the trial I'm discussing is sponsored and funded by Zentalis, who produces and developed ZN-c3. So my talk today is going to be split into two different parts. The first is really going to go into some of the background and biologic rationale behind the trial itself. And then the second half will actually focus on the logistics of the clinical trial and what it looks like to be on the trial itself. So if you will humor me, we're going to take a little trip down memory lane back to high school and college. And I recognize for some of you that maybe now. And I will very briefly go through the cell cycle and the checkpoints and some of the key proteins, as well as some of the preclinical data that form the foundation for this clinical trial. So as you may recall, the cell cycle is a very tightly regulated process. It helps govern how our cells divide and grow over our entire lifespan. And during that lifespan, they encounter a lot of stresses, both from within and from outside the environment, so endogenous and exogenous stressors. And so during the 4 different phases of the cell cycle, there's different parts that are responsible for helping heal those, the damage to the cell, and specifically damage to the DNA. And the underlying machinery of that DNA really takes effect during certain checkpoints. So I'm specifically going to talk about the G1S checkpoint and the G2M checkpoint. And I promise it won't be too much detail, but this is the DNA damage repair pathway from a very 30,000-foot view. So on top, you'll see the blue double helical structure of our DNA, the parts of our genes. And over time, the damage that occurs will cause either nicks, mismatch between pairs, or even actual breaks through the entire strand. And the cell's machinery has developed about 4 different pathways that it can take to repair this, and which pathway it takes will really depend on the type of damage that has occurred. For the purposes of the talk, I'm really going to focus on what's called the homologous recombination pathway, or the HR pathway, and that's because that pathway is what really contains p53 and the Wee1 kinase. So osteosarcoma, like a lot of sarcoma, unfortunately, can become quite smart and resistant to a lot of the things we do to it. And one of the ways it does that is, as over time, it really starts to have mutations in p53. And depending on the time frame at which biopsies are done, about 90% of all osteosarcomas have a mutation in TP53. And that's important because P53, which is the guardian of our genome, is responsible for sensing any DNA damage that's occurred and forcing cells to take a moment to repair and heal. And if P53 is mutated, it's unable to function in that role, and then the G1S checkpoint really becomes non-functional. And therefore, cells really depend on the other checkpoint, the G2M checkpoint, to help take time to heal themselves so that they don't continue to accumulate DNA damage and undergo this process called mitotic catastrophe, where they just become too unstable and then die off. So the G2M checkpoint is regulated by a protein called the Wee1 kinase that in turn controls a lot of different proteins that I won't go into. But the thing is, when Wee1 kinase is activated, it causes cells to pause and recover any damage that's occurred. Osteosarcoma, because the G1S checkpoint has been obliterated, it really is not functional. It relies on this G2M, and the way it does that is actually ramps up the amount of Wee1 kinase that's present so that there's a higher risk that cancer cells will take a moment to heal themselves. And so the theory is that if you use an inhibitory molecule or drug against Wee1, you prevent that healing time to occur, and therefore cancer cells will continue to accumulate more and more DNA damage and will eventually die off. So in the top row, that's the idea of just Wee1 inhibitors by themselves. More DNA damage over time, eventually they die off. The bottom row, on the other hand, is looking to prime cancer cells with DNA damage, so things that everyone already unfortunately has to go through: chemotherapy, radiation therapy, those kind of things. And then instead of allowing those cancer cells to recover, you force them to undergo mitosis and divide and hopefully die off. So this kind of rationale was proven in preclinical studies of osteosarcoma cancer cell lines, as well as mice studies. And so you have two different figures, one on the left and one on the right. I think that the key things I want to highlight is that Wee1 inhibition just by itself was effective regardless of what the P53 status was in various osteosarcoma cell lines. And then the other key thing was that the ranges at which these drugs seem to be effective was in the therapeutic range that we expect for drugs that are viable in human clinical trials. And then to the second part, which is that Wee1 inhibition, when primed with other DNA-damaging agents such as gemcitabine, which causes double-stranded breaks, really seems to make that effect even more. And again, irrespective of P53 status. So on the right-hand side, you'll see in blue is cancer cells that are not exposed to anything. In green is when they're exposed to just gemcitabine. In orange is when they're exposed to Wee1. And then the red line at the very bottom is when they're exposed to both. And so there's more and more effect as we go through that kind of process. I'm going to shift gears real briefly and talk about the actual logistics. So what does the study look like and what can a patient expect to experience when they're on this trial? So this is a phase 1/2 study. It's a dose escalation, dose expansion study. So it is the first in human of this combination of drugs. And so the phase 1 is the dose escalation and safety lead-in. And we plan to enroll 12 to 18 patients, and this is actually going to likely go to 24 patients. The phase 2 is the dose expansion and the efficacy, and we plan to enroll approximately 60 patients. And in order to enroll onto the trial, patients do have to be older, or 12 years or older, and weigh at least 40 kilos, roughly 88 pounds. And the reason we have those requirements is it depends really on the way that the drug is administered as opposed to anything about the biology of the tumor itself. Again, this is just the real layout, and the one thing I want to bring your attention to is that here we have multiple check-ins during both the phase 1 and phase 2 portion. Because this is such an investigational therapy, we want to make sure that we're not seeing bad side effects, or that we are seeing hopefully some type of signal so that we're not putting patients on something that we're worried may not be working or is too dangerous. So the study objectives are pretty standard. The phase 1 portion is really looking at, are there any major side effects or what we call dose-limiting toxicities that require a pause or even coming off of a trial because of how severe they were. And during that phase 1, we also identify the dose that we take to the phase 2 portion, what we call the recommended phase 2 dose. And the phase 2 is really looking at, is this treatment combination effective? And the way we do that is looking at event-free survival, or EFS, at 18 weeks. And that's based on a benchmark that the Children's Oncology Group set with all of our historical phase 2 trials. There's also a couple of exploratory and secondary aims, and these are getting more at like what happens at 12 months. how do patients actually feel when they're getting this therapy? And then I think somewhat very interestingly, we're looking to see, if there is someone who is responding, why are they responding? Is there something unique about their tumor that we can better understand so we can better guide our therapy towards patients who have similar types of changes? Treatment is based on cycles of 3 weeks, so 3 weeks or 21 days. During that time period, ZN-c3, which is an oral pill, it's an oral WEE1 inhibitor, is taken once a day, every single day. Gemcitabine is an IV drug, so either given through a port or through the IV in the arm. That's dosed on days 1 and day 8 of that 21-day cycle. And every 6 weeks, so every 2 cycles, we get scans, either a CT or an MRI, to see the status of where the tumors are and how big they are. The inclusion, so in order to be on this trial, it's pretty standard. You have to have measurable disease by a radiographic criteria, what we call the RECIST version 1.1. You have to be healthy and strong enough because, again, we don't know what necessarily all the side effects will be, although we have an idea. So we want to make sure people are going to be safe. You have to swallow a pill. Unfortunately, as it stands right now, it cannot be crushed. And then for patients over the age of 18, we do require that they get biopsies on treatment. Again, this is really to help us understand if they're benefiting, why they may be benefiting, and if they're not, what could be unique about their tumor that's preventing such a response. And then as with all investigational therapy, you have to be on contraception. And then importantly, you can have had any number of prior lines of therapy, including with gemcitabine. Potential side effects, this is relatively not unique to this combination. This is the most chemo and targeted therapy, so people may feel tired, their blood counts are going to be lower, there can be inflammation in the liver, and then you can have an upset stomach, nausea, throwing up. And then there are some exclusionary criteria, but again, this is relatively standard. If you've been on prior therapies, we want to make sure that there's no overlapping toxicity, so we want to wash that out. You can't have active brain metastases. We want to make sure your heart and your gut are strong enough, and you can't have had a prior WEE1 therapy. So these are the participating sites, and this is actually outdated as of a few days ago. We now also have the Fred Hutchinson Center who has opened this trial, and we're actually planning on opening this at other institutions as well. And this will become an international study. We've already received approval from the countries listed here. There's a lot of people who've made this possible, so I won't take time to go through all of them, but I do want to thank all the patients and families, because without your support and without your faith, none of us can do what we do. So thank you.
Hello, everyone. Thank you to the MIB team for inviting me. I'm really excited to be here and talking about this trial. I'm Laura Davis. I'm from the Knight Cancer Institute at Oregon Health and Science University, and I'm excited to tell you about our trial, which is in progress, SARC-38, which combines the oral multi-targeted inhibitor regorafenib with the immune checkpoint inhibitor nivolumab for the treatment of metastatic osteosarcoma. I'm pleased to be presenting on behalf of the whole study team, most notably my co-PI, Dr. Fariba Naveed from CHLA. SARC-38 is a phase 2 trial that's sponsored by SARC, which hopefully many or all of you have heard of. It's a large national research consortium focused exclusively on sarcoma research. So as many of you know, thus far, immunotherapy for osteosarcoma has not been quite as effective as we had initially hoped. We've heard a lot about that over the past couple of days. In 2015, SARC opened a trial of one of the early immune checkpoint inhibitors, pembrolizumab, and SARC-28 was really a signal-finding study, which was using pembro to treat a bunch of different kinds of sarcoma subtypes. And through this trial, we discovered that pembro can be quite effective for undifferentiated soft tissue sarcomas, but really not very exciting for osteosarcoma. This graph, which we as oncologists look at all the time, but just in case you're not familiar with this kind of graph, it shows time in weeks along the horizontal axis, and then along the vertical axis, the percentage of patients who did not have disease progression. And for patients with osteosarcoma, SARC-28 suggested that the average time to tumor progression is the same for a patient on pembrolizumab as it is for a patient on placebo. However, there were a few patients on this trial that clearly benefited, including one patient that continued on pembrolizumab alone for over a year with tumor regression, and multiple other patients that had stable disease. And the percentage of patients that were progression-free at 4 months seems to suggest at least some activity of immune checkpoint inhibition, even by itself as monotherapy. And then a couple of years after SARC-28, we really finally had a breakthrough. Patients with metastatic osteosarcoma treated with regorafenib have a definite improvement in disease control. So regorafenib is an oral targeted small molecule inhibitor that targets many different growth signals, cancer growth signals, especially one called VEGF, as well as many others. And regorafenib more than doubles the time of osteosarcoma disease control. So as a result of this trial, regorafenib is now included in national treatment guidelines for the treatment of metastatic osteosarcoma, which honestly is mostly important because then insurance companies need to cover the treatment. And in the future, we're thinking about maybe studying this sort of treatment to help patients remain disease-free after, for example, lung metastases are surgically removed. And while we're all in the community very thankful that we actually have a new treatment that works, we also completely recognize that 3.6 months of disease control is not good enough. So we know that immune checkpoint inhibition, like with pembrolizumab or nivolumab, it's not effective enough on their own. And we believe that this is because there's really not enough immune cells infiltrating osteosarcoma tumors, and that the few immune cells that are there are just not doing their job. And then also, we now know that multi-targeted tyrosine kinase inhibitors like regorafenib or cabozantinib are effective, but certainly not a curative treatment, not effective enough. Importantly, these kinds of small molecule inhibitors also have a really important but unappreciated impact on the immune system. And I'm going to show this rationale for a combination in a very simplistic manner, so forgive me, but hopefully these cartoons will kind of walk you through some of the basics. A normal, healthy bone is very organized, and it's very structured with normal blood vessels. But when a bone cell becomes cancerous, that begins to change. And if that cancer cell is not destroyed right away, it replicates and becomes an osteosarcoma tumor, which can then spread and metastasize. And these tumors are also characterized by this really abnormal blood vessels that with very few immune cells, particularly very few T cells. So there's a few immune cells within the osteosarcoma tumors, but those immune cells tend to be helping rather than attacking the cancer cells. There should be activated T cells in the tumor attacking and destroying cancer cells, but in osteosarcoma, the rare T cells that are there really that make it into the tumor are inactive. And that's in part due to VEGF, which is a protein produced by the osteosarcoma cells, as well as the macrophages in the tumor. And VEGF is also a major reason why the tumor blood vessels are all messed up. So inhibition of VEGF with a drug like regorafenib allows the blood vessels to normalize and allows more immune cells to come into the tumor. But the tumor still has other tricks that make it avoid the immune cells. And that's where a drug like nivolumab can help, because nivolumab helps reveal the cancer cell to the T cell and trigger the T cell to activate and attack the cancer cell. So, very simple, but simplistic. The process is much more complicated, but combinations like regorafenib and nivolumab have been actually very successful in some other cancers. And so, for all of these reasons, SARC-38 is bringing it to osteosarcoma patients. So SARC-38 treatment involves both regorafenib and nivolumab. Regorafenib is a pill. It's taken once a day at home for 3 weeks out of every 4 weeks. You get a week off. Nivolumab is given IV at the study site. And for kids, it's given every 2 weeks. And for adults, it's given every 4 weeks. And a really beautiful part of this trial is that after 4 months, if you have disease control, but you have something that could be cut out, you can get it cut out and remain on study. You can go to surgery, recover from surgery, come back and resume treatment if you want. You don't have to. For this trial, the eligibility includes anybody over the age of 5 that can swallow intact pills. And you have to have osteosarcoma, of course, at least have received 1 line of MAP, chemotherapy generally, and overall be doing fairly well and have that magical resist measurable disease, which essentially means something in your lung that's more than 1 centimeter or something along those lines. No prior regorafenib or cabozantinib or similar kind of drug, or no prior nivolumab or similar type of drug. So SARC-38 is designed in 2 stages, and since this is a trial that's currently in progress, I really cannot discuss results at all, but I can share with you that we have enrolled 12 patients thus far, and the first patient enrolled about a year ago and remains on study treatment, and multiple patients appear to be receiving some degree of clinical benefit. We very much want this trial to benefit any patient who enrolls, but we also want to make sure that we learn as much as possible so that we can make it better for the next patients too. And so we're collecting tumor tissue, blood samples, and stool samples, and collaborating with researchers across the country, many of whom are in this room, to study those samples. And a tumor biopsy after 3 weeks of treatment on study is required for adults enrolled on this study, and it's optional for kids. And I want to just take a moment to really emphasize that while, for example, studying circulating tumor cells and circulating tumor DNA holds a ton of promise, there are aspects of osteosarcoma that just cannot be understood or studied without the intact architecture of the tumor microenvironment from biopsies. So I really want to encourage all patients on clinical trials, any clinical trial, to please consider providing a tumor biopsy. It will absolutely be an invaluable resource that will advance the field. Thus far, we are open at these sites, and we have multiple additional sites planned. We made a very conscious effort to open this trial at geographically diverse sites in hopes that this trial can be accessible to as many patients as possible. And with that, I want to say thank you, and I'm happy to answer questions at the end.
All right, thank you so much. I just want to thank the organizers for this opportunity to present to you some of the data from our prospective clinical trial, where we have been evaluating the effects of a highly attenuated listeria that expresses a recombinant HER2/neu molecule to prevent metastatic disease in dogs with spontaneous osteosarcoma. And so I do have a disclosure. I am an inventor on a patent for this vaccine, so please bear that in mind. So why listeria? I think probably you've heard of this. It causes food poisoning on the cruise ships, right? Why would we be using that type of approach to stimulate an anti-tumor immune response? And I think when you look at sort of the biology of listeria, it becomes obvious, hopefully, as to why we're doing that. Listeria is a really potent inducer of innate and adaptive immunity, and that's exactly what we're trying to do. We're trying to stimulate immune response against HER2/neu. So if you sort of follow along in the cartoon here, listeria is in the green. Listeria monocytogenes, that's its name. It likes to go to mononuclear cells, which are antigen-presenting cells. So when we have a genetically engineered Listeria that has been modified to express HER2/neu, we can use Listeria to take that HER2/neu directly into antigen-presenting cells. When it gets into those cells, it goes into the phagosome and it starts to get digested. And so products, Listeria products, as well as the HER2 it's carrying, will actually be presented on the surface of that antigen-presenting cell in the context of MHC class II to stimulate CD4 positive T cells, helper T cells, against both Listeria and also HER2/neu. But the real key thing about Listeria is its ability to secrete this molecule called Listeria lysine O. That is a pore-forming protein, and it enables Listeria to escape from the phagolysosome into the cytosol. And there it gets processed to be presented on the surface of these antigen-presenting cells in the context of MHC class I. So now we can get a cytotoxic T cell response against HER2/neu. In addition, Listeria is a really potent inducer of cytokines, and that's going to become important later on. And it does that through activation of both NF-kappa B and the STING pathway, which we heard about earlier as well. So basically, we're able to use Listeria as a vector to deliver HER2 directly into antigen-presenting cells and stimulate a potent CD4 and CD8 T cell response and cytokine response. So we wanted to ask the question, could we use this Listeria to prevent metastatic disease if we used it after standard of care therapy in dogs? And that's amputation and chemotherapy. So that was the first thing we wanted to know. And then the second thing we wanted to know is, are there correlative biomarkers which could tell us which dogs might respond to this therapy and which dogs may not respond to this therapy? So we set up a fairly simple protocol where dogs would be evaluated for osteosarcoma, confirmed to have the diagnosis, undergo amputation, 4 doses of carboplatin, and then if they were free of metastatic disease at that point by thoracic radiographs, they would be injected with the Listeria, 3 doses of Listeria only. And then if they made it out to week 23, that was basically then the group of evaluable dogs. And so we compared that with a standard of care arm that had been previously enrolled in a previous clinical trial. Everything about those dogs in the standard of care was exactly as I described for the vaccinated dogs, except for they did not have the vaccine. And so you can see our standard of care arm here and our vaccine arm in terms of intent to treat 149 dogs versus 118 dogs. And then you can see in terms of free of metastases, 63% I think it is compared to 68%. And then the evaluable dogs that make it out to 23 weeks, we see 56% compared to 64%. Why were we excited about this? This is published data, and these are the results from our initial study where we did exactly the same, but this time we were using or selecting dogs that expressed HER2 in their tumor. In the national study, we didn't look at that yet, and they were treated with 3 doses of vaccine and received boosters, which again, these dogs did not receive. So in terms of the outcome, what do we see? Interestingly, we saw no difference in the disease-free interval between those dogs that were treated with standard of care versus Listeria, but we did see a statistically significant increase in overall survival, suggesting that even if they did develop metastases, they were able to survive longer following the vaccine. However, when we looked at the evaluable dogs, so those are the dogs that made it out to week 23, we did not see a statistically significant increase in either disease-free interval or overall survival. And we're looking at this now to look to see which patients actually do express HER2 and whether that actually, if we stratified them according to HER2 status, whether that would be important and change things. But what was important is we could now look at these dogs and ask, okay, we've got a group of dogs that did well, and we've got a group of dogs that did not do well that received the Listeria vaccine. So that was 75 evaluable dogs. And we were able to take the top 25, those that did very well, had prolonged disease-free intervals, and compare them with the bottom group of dogs that had received the 3 vaccines but did not do well. And the first thing we were able to see is at the time that these dogs received the first vaccine, there was a difference between these two groups. The dogs that went on to have long-term survival had a statistically significant increase in temperature 4 hours after the first vaccine, whereas the dogs that did not have prolonged survival did not show that response. Furthermore, when we started to look at the cytokines, we could see a clear difference. So each of these graphs looks at a particular cytokine in the serum of these dogs before vaccination and 4 hours after the first vaccination. And hopefully what you can see is when we looked at the long-term survivors, they were able to show a statistically significant increase in their cytokines at 4 hours. But the dogs that went on to do poorly did not have that increase. So clearly at that time point, there's something different immunologically between these two groups. We also found, when we looked at some other cytokines, IL-6 being an important one, is that the poor responders could actually increase those cytokines, but the magnitude was much less. So again, it tells us that the innate responses of these dogs is different and could potentially be used as a correlative biomarker, essentially of immune fitness. And then when we looked at using gene expression profiling in nanostring, after the third vaccine, we also could see a difference in the gene expression profiles of the peripheral blood mononuclear cells between those dogs that were long-term survivors versus those dogs that were short-term survivors. So in those dogs that went on to do well following vaccination, we could see upregulation of effector molecules like perforin and granzyme, some CD8 T-cell differentiation factors, some NK responses as well. And so that suggested that following vaccination, that group of dogs were able to upregulate what looks like a cytotoxic T-cell/NK cell response, whereas those dogs that went on to do poorly did not show that response several weeks after the last vaccine. So I think this brings up an important point when we're thinking about immunotherapies, which is I think we should be thinking, in addition to all the other things that we're thinking about, we should be thinking about immune fitness. We should be thinking about the ability of the patients to be able to respond to the immunotherapy. How fit are they? And if they are fit, perhaps they're going to respond better to immunotherapies. And I think this is borne out by the fact that we can see there are clear defects in monocyte macrophage activation early on in this group of dogs that went on to do poorly. And there are also defects in their T-cell responses. Now, whether that is because of the early defects in innate immunity that's preventing those T-cells from getting appropriately activated, or whether there's intrinsic defects in the T-cells themselves, is something that we need to start to tease out. But also, I think, just to add, I haven't had time to show that, but the vaccination in these dogs appeared to be safe, just mild transient side effects of fever in dogs that went on to do well, and some nausea and some vomiting. And in that intent to treat group, we did see a statistically significant increase in survival. But again, that wasn't seen in that evaluable group, and we need to do a lot more interrogation of the tumor to really try and understand that a little bit better. But I think that what we would like to do is really understand these defects early on. And could we actually use this to actually test dogs early on and say, are you fit? Are you able to respond to an immunological stimulus? And if you are, then you can go on and receive your immunotherapy. And if you are not, we have to understand why you're not, and could we turn you into an individual that could potentially respond and could then potentially go on and have a positive response to your immunotherapy. We also have to look, obviously, at HER2 stratification and ask that question about repeat dosing as well. So lots of people to acknowledge here: the clinical team, the Comparative Oncology Trials Consortium at NCI, obviously all the trial teams across the country, and of course all the canine patients and their families. So thank you.
I think we have Rosie Kaplan next for virtually. I don't know if someone back there has her.
Are you good? We got you.
Can you see everything when I change the slides? Perfect.
You're on screen, go.
Hi, everyone. Hope you can hear me and see me okay. It's Rosie Kaplan. Sorry not to be there in person. This is one of my absolute highlights of the year meetings. I feel really honored to have seen these meetings grow, and it's near and dear to my heart, so I'm sorry I'm not there in person. But hopefully we can enjoy some discussion throughout the way the world is now, virtually or in person eventually, hopefully. Today I'm going to talk about the immune environment in osteosarcoma and our kind of collaborative efforts to develop next generation immune cell therapies. And I just want to start off by saying that this is a very collaborative work that I'm sharing, that crosses many years and many people, and hopefully I'll have time to give everyone thanks to that. But there are definitely many people behind all of these slides, including my lab and then the clinical teams, both here and now at Stanford, and many other sites that are soon to be opening and involved in all of this. And so it's very collaborative in that way. And hopefully I'm not too redundant to what's already been talked about in the meeting. I apologize for that if that's the case. So just a little background on the osteosarcoma immune microenvironment and our interest in metastasis. Osteosarcoma forms in the bone, but can often metastasize to the lung, and sometimes to even other bones. And my laboratory is very interested in the environment and how it changes both in the lung and the bone when osteosarcoma metastasizes. And so we've been very focused on this tumor microenvironment and the process of cancer spread. And one thing that we've found is, in addition to the cancer cells themselves, which are, we think, especially in osteosarcoma, are very unique, and each patient has very unique genomic features. But those genomic features that are unique to that patient then have unique microenvironmental interactions, and those interactions are, I think, very, even more, and maybe I would say, dare to say, even more interesting in osteosarcoma than and other cancers that metastasize. And many cancers do have the process of metastasizing or spreading. But we hope that understanding this crosstalk, this interaction between immune cells, stromal cells, blood vessel forming cells, and the tumor cells themselves can help us understand the process of cancer spread and allow us, most importantly, which my lab has been predominantly focused on, developing new therapies based on that understanding, and how we kind of interact or intercept this kind of what we think is essential crosstalk between all the cells that are normally in your organ and recruited to the organ when you have metastasis. And each cell actually plays, each cell and compartment plays unique roles, and even non-cellular structures such as stroma and extracellular matrix can also play really important roles. And so hopefully in the years to come, I'll talk about different aspects. There are people in my lab that are looking at the extracellular matrix in this process, collaborative efforts looking at the endothelium, but predominantly in this talk and many focuses in my lab is on the immune compartment, and in particular harnessing T cells, which are known to attack and kill tumor cells, and how to best use these as they're interacting with other cells. And that's basically the focus of my talk today. So we've been very interested in the osteosarcoma immune microenvironment, and we're now in the thick of a manuscript. This is all unpublished work, but hopefully will be published soon, where we're trying to really understand patient osteosarcoma samples compared to osteosarcoma mouse models. So this is a mouse model, and unlike many mouse models that we see for osteosarcoma, this isn't human osteosarcoma, it's mouse, and it's in an intact mouse with an intact immune system. So the mice has all the immune components. And when we look at these tumors that are in the, that are derived from a genetic alteration with a p53 knockdown, and thanks to Jason Eustein for the cell lines that we've now modified. They're metastatic and then they're traceable. And when we look in these tumor models that are formed in bone over time, we see the predominant cells are myeloid cells, monocytes, macrophages, and neutrophils. These cells are very important in the process, and we see very few T cells, and those T cells there are exhausted. So that led us to think, okay, we need to do something to get T cells in and have them be functional. So we wanted to increase functional cytotoxic T cells in individuals. In order to do that, there's a process where we would take their T cells, change them, and bring them back into the host, and that's called adoptive T cell transfer. And we found that GD2, which is a fat protein molecule that is found highly abundant on osteosarcoma, human osteosarcoma cells, as shown here, as well as neuroblastoma. And this is a metastatic osteosarcoma in the lung, and you can see really very little staining in the normal lung and lots of staining in the tumor. And so the first trial we did at the NIH was a GD2 CAR trial, and we engineered a chimeric antigen receptor, which has an antibody component and a signaling component, and also has a insertion for a gene that will allow the chimeric antigen receptor to not signal and to kill the cell in case of toxicity. The trial we did was to look if we could feasibly make these cells, take T cells out, manufacture them, by engineering them to express GD2 so that they would kill GD2 expressing tumor cells. And we did this in patients with osteosarcoma and neuroblastoma, and we treated the patients with cyclophosphamide as a conditioning regimen. What we found was very good expansion in some patients and poor expansion in others. And we're really trying to understand more about that. And it wasn't just based on dose, whether the patient had a good expansion or not, and we're actively looking into that. We were able to enroll 15 patients, age 18 to 28 years of age. There were no dose-limiting toxicities found. We were able to get up to the highest dose of 1 × 10⁷ per kilo of CAR T cells, and we found that it was safe and feasible. The patients only had 15% experienced grade 1 cytokine release syndrome, but there was no neurotoxicity and overall very well tolerated. We are now actively involved in understanding the good versus poor expanders. The good expanders we see have a large proponent of NK cells in their apheresis compartment, whereas the poor expanders have a large component of these immune suppressive monocytes. And when we look at the CAR product itself, we find that the good expansers have active non-apoptotic CD8 cells, whereas the poor expanders have more apoptotic cell expansion. And so we felt that maybe with this trial, that one of the reasons that was a problem was that maybe the T cells themselves were getting exhausted, and maybe we could prevent that and improve the manufacturing. So Crystal Makel's team developed a culture system with dasatinib, where this would kind of pause the T cells in this quiescent state and allow them not to be signaling and activated. And you can see here they express much less activation markers when they're and exhaustion markers of LAG3 and TIM3 when they're cultured with dasatinib. And when those cells are put back into mice, they actually kill tumor better. And so now we have a new trial called GD2 CAR Persist. We think if the T cells are less exhausted and less activated, that they will persist longer. And so we now have a new trial open where we would give conditioning not just with cyclophosphamide, but fludarabine, which is shown to be very important in getting the T cell milieu to be ready to accept the CAR T cells and to foster their expansion. And we are actively enrolling. So we have a new engineered CAR that has a CD8 transmembrane domain and a 4-1BB co-stim, which we think will add to the persistence along with the dasatinib. And we are actively screening patients. We would give them lymphodepleting chemotherapy, the infusion, and then watch them closely for toxicity. And we're open at the NIH, actively enrolling patients, and we will be open at multiple sites soon. This is a multi-site trial. And the exact same CAR, GD2 CAR, is is being manufactured in the exact same fashion at Stanford and has shown to be with very promising and exciting results in diffuse midline gliomas that also express GD2. So these tumors also express GD2, and the same CAR product had some very nice preliminary results in the first 4 patients. This is a picture from their Nature paper. You can read about this. And so we're very excited to have kind of parallel correlative studies to compare patients with osteosarcoma, neuroblastoma, and diffuse midline gliomas being treated with this CAR. And so I'm not sure how much time I have left, but hopefully I didn't go too far over. I really want to thank Melinda Merchant, who was hugely involved in the first trial and the clinical work, and Crystal Makel and Sneha Ramakrishna, who's actively involved, as well as Robbie Major in the DIPG trial and our current osteosarcoma neuroblastoma trial, and Kristen Wessel in my lab, and Sabina Kauchanowska also helpful, and of course all the patients and their families that make such a difference in all of this and motivate me to do what I do every day.
We're going to move forward. Hopefully everybody's got ice cream or guacamole or something to keep them going. Dr. Healy is showing off his ice cream. All right, so Katie.
All right, great. So everybody should be happy then. They have ice cream and snacks. My name is Katie Albert again. I come from Seattle Children's and the University of Washington. I'm really excited to be here. It's my first Factor conference, and like everyone else, I'm super impressed with this organization and the conference, and really happy to be in a place where researchers and patients and families are coming together. It's a really cool way to talk about the work that we do. So I'm going to go ahead and get started, and I'm going to, kind of as a good follow-up to Dr. Kaplan's talk, I'm also going to be talking about CAR T-cell therapies in sarcomas, and hopefully I can piggyback a little bit on some of the foundation that she laid for us. I don't have any disclosures. This is an overview of what I'm going to talk about. I'm going to give a very brief now introduction to what CAR T-cell therapy is. Talk a little bit about the challenges also mentioned by Dr. Kaplan for sarcomas and solid tumors, and then briefly review the CAR T-cell trials that we offer at our institution. So again, a CAR is a chimeric antigen receptor, and it's an antibody fragment that is connected to T-cell signaling machinery. And CAR T-cell therapy happens you can see in the diagram to the right here, when we collect T cells from patients and genetically engineer them to express this CAR. And we select antibodies against tumor antigen targets. And the idea is that then when we reinfuse those cells, they will find the tumor cells, they'll kill the tumor cells, and they'll release a number of other immune molecules called cytokines that are also anti-tumor, thus kind of waging an immune war on the tumor. CAR T-cell therapy has been quite successful in a subset of leukemias and lymphomas, but we're really just getting started in trying to figure out how to best use CAR T-cell therapy in sarcomas and solid tumors. There are a few really clear challenges for us, including the following. In leukemia and lymphoma, you typically have a lot of circulating tumor cells that have the tumor antigen uniformly expressed, and in most cases where CAR T-cell therapy has been effective for leukemia, it's also on targets that are present on normal lymphocytes. So the CAR T-cells interact with their target right away, and that activation and expansion can happen right away. In solid tumors, there may be some circulating tumor cells, but really the CAR T-cells have to traffic to the sites of bulk tumor disease. So there's a whole journey that has to happen after infusion. In addition, there's not uniform expression of tumor antigens most of the time in solid tumors. So there may be some degree of antigen density that has an effect on how well the CAR T-cells work. And as Dr. Kaplan was getting into, when you have these bulk tumor sites, you have this kind of power in numbers of the tumor cells that produce this immunosuppressive tumor microenvironment. So CAR T cells against solid tumors have a few really big challenges that we have to keep in mind as we're designing therapy for our patients. I'm part of a team at Seattle Children's with Julie Park and Naveen Pinto, who have spent the last several years designing these trials. And our focus is always on developing therapies for the highest risk patients, the greatest need patients, and of course, continuing to try to find better, less toxic therapies. First, I'm going to review our solid tumor basket CAR T cell trials, which are called STRIVE, which stands for Solid Tumor Immunotherapy Engineered. It was a stretch. I didn't come up with it, but it works, I guess. And we currently have 2 of these basket solid tumor trials open under the STRIVE title. And they follow the same general format, which is that they are both phase 1 studies, so we're evaluating safety, feasibility, and in both cases, patients come, undergo apheresis, have manufacturing of their CAR T cells, can get some treatment while we're manufacturing their CAR T cells, then they come back, they get lymphodepleting chemotherapy, similar to what Dr. Kaplan described with cyclophosphamide and fludarabine, then they receive their CAR T cell infusion, and we observe them for at least a month. Also, both of our trials under STRIVE have 2 arms. The first is simply a dose-finding arm, Arm A, for the tumor antigen-directed CAR T cells, and then Arm B, which occurs subsequently, which is a bispecific dual transduced CAR T cell product against the tumor antigen and CD19. CD19 is expressed on normal B lymphocytes and therefore in some leukemias and lymphomas, and in fact is the target that's been quite successfully targeted by CAR T cells. In this case, we're not using CD19 as a solid tumor or sarcoma target. We're using it to try to promote the expansion and persistence of CAR T cells so that we can get those CAR T cells to their target and keep them in high enough numbers that they can overcome the immunosuppressive tumor environment. Our target in STRIVE-01 is EGFR, which is the epidermal growth factor receptor, a very important protein for cell growth and differentiation, both in tumors and in normal tissues. EGFR has been implicated in many pediatric and adult solid tumors and has been targeted with antibodies and other therapies, but the problem is that there's limits to how well they can work because there's so much on-target, off-tumor toxicity. What got us really excited about EGFR is that there's an antibody that binds specifically to tumor-specific EGFR, because when EGFR is overexpressed in tumors, it folds in a different way. So the EGFR 806 antibody binds in a pocket that's only available when the protein is in this untethered folding pattern and not when it's in its normal folding pattern in normal tissues. Most of the patients that we have screened for this trial have not had high enough EGFR expression that they were eligible, but about 25% do, so there is a screening step to the study. And then the pie chart shows just the distribution of the histologies that we've seen in like most pediatric and young adult phase 1 solid tumor studies. It is highly enriched for sarcoma, including osteosarcomas that have anywhere between a 35 and 40% EGFR IHC positivity rate. Very quickly, we've actually completed Arm A of this study, and it was well tolerated with the exception of one dose-limiting toxicity of an immune-mediated hepatitis. We decided to move on to Arm B because we were seeing some signals of activity in a few patients with mixed responses. We are seeing expectedly low level engraftment compared to what CAR T cell trials in leukemia and lymphoma patients show. But again, we have seen some mixed responses, meaning some tumors remaining stable or getting bigger and some tumors getting smaller. And the pattern we've seen in 3 of our patients is that larger tumors tend to have more reduction and smaller tumors tend to be stable or grow bigger. We've seen these mixed responses in 2 soft tissue sarcomas and 1 osteosarcoma. STRIVE-02 is our second STRIVE trial, and in this trial, B7-H3 is the tumor antigen target. This is a a protein that's important both as an immune checkpoint molecule and for cell growth and invasion, and is very uniformly and highly expressed on most sarcomas, so that's what makes it a really attractive target. And my colleague, Dr. Naveen Pinto, is the PI of this trial. It has also completed enrollment on Arm A and is moving on to Arm B. It was also well tolerated with relatively low engraftment, and there weren't any of those promising responses except in a patient that had stable disease and got a second infusion, had very high engraftment, which I'll show you on this next graph. It's the magenta line, and actually had a pretty remarkable PET response. Of note, that patient also had a dose-limiting toxicity of immune-mediated hepatitis. And then finally, and probably of interest to this group, we just opened another CAR T-cell trial for osteosarcoma patients in particular called Enlighten. And a lot of the logistics of the trial are similar in terms of undergoing apheresis and the manufacturing, and the DLT monitoring period. But in this case, the CAR is not against a tumor antigen that's on the cell surface. It's against this CAR T-cell adapter molecule that is being made by the company Umoja, which is a fluorescein or FITC molecule that's conjugated to folate. As some people here probably know, folate receptors are overexpressed in osteosarcoma. So in this case, the CAR binds to the CAR adapter molecule with the expectation that it will bind to the tumor cells. The other difference to the study is that we give several escalating doses of that CAR T adapter molecule so that we can basically find a max tolerated dose for an individual patient, and the patient can go on to receive several other infusions if they're doing well. So in summary, it's an uphill battle, but we're dedicated to finding the answers and overcoming these challenges, and really thankful for patients and families that trust us to take care of them on these trials. Thank you.
Hi. Hello, everyone. I'm Bhuvana Sari. I'm from Nationwide Children's Hospital, and I'm going to stick with the immunotherapy theme, but switch concepts a little bit to talk about natural killer cells for osteosarcoma and other sarcomas. So this work is really built on a lot of the basic science work that has been done by my collaborator, Dr. Dean Lee, who I know has attended some of these previous meetings, and you may have heard some of his preliminary data on the preclinical side. So I'll talk a little bit about the background and where we are in the trial development at this time. So just as a refresher, what are natural killer cells? These are regulatory cells that are engaged with dendritic cells, macrophages, T cells, and other endothelial cells. And they're basically effector lymphocytes of innate immune systems that control several tumors and microbial infections as well by limiting the spread and subsequent cytotoxicity of cells. There's a lot of preclinical data and evidence to suggest that NK cells can be used as an adoptive anti-sarcoma model, and they've shown significant cytolytic activity against pediatric sarcoma cell lines from many histologies, including osteosarcoma. We've heard a lot about the tumor microenvironment and how that is difficult and something that we have to overcome in trying to help our immunotherapy trials be successful. So one of the things that was worked on in Dr. Dean Lee's lab was to figure out how to protect the NK cells from the tumor microenvironment. What they realized is that when they exposed NK cells to TGF-beta during the expansion process, it hardens them against later exposure within the tumor microenvironment. And this process is called TGF-beta imprinting. So in order to figure out whether this imprinting process has significant effect on other activities of the NK cell, various experiments were done. They identified that there was no significant effect on fold expansion of the NK product and no effect on the expected viability. In fact, re-exposure to TGF-beta generally reduces degranulation in typically expanded cells, but this function is also preserved in TGF-beta imprinted cells, and the cytolytic activity against sarcoma cell lines is also protected. So one of the big problems with immunotherapy trials are the occurrence of cytokine release syndrome, which is basically a syndrome that can be associated with fevers, headaches, nausea, sometimes hypotension, and even respiratory difficulty. So looking at TGF-beta imprinted cells, what was identified was that there were enhanced cytokine secretion compared to typically expanded cells, and this enhanced cytokine secretion is usually preserved upon re-exposure to IL-2. However, importantly, IL-6 is really not a prominent cytokine of either expansion product, so we expect that the cytokine release syndrome should be of low concern in patients when this product is used. So this is a very interesting recent study done in canine osteosarcoma by Dean Lee and some other vet school colleague, where it was a phase 1 study of universal donor TGF-beta imprinted NK cell therapy that was used in combination with carboplatin for canine osteosarcoma. This was a phase 1 trial, basically to look at the safety, toxicity, and feasibility of off-the-shelf NK product in canine patients with osteosarcoma. It was done as a dose escalation study with a 3+3 study design to look for and to determine the maximum tolerated dose. 4 infusions of perioperative NK cells were followed by 4 cycles of carboplatin chemotherapy every 3 weeks, with 2 infusions of weekly NK cells after each dose of carboplatin for these canine patients. So what you can see is that 3 dogs completed the infusions at dose level -1 and dose level -2, and among all the canine patients, they received a total of 54 NK cell infusion. Out of the 5 that were initially studied in this cohort, 4 of them showed no lung metastases, but 2 developed metastases at different sites. And the only toxicity that was noted was an acute infusion reaction that was eliminated by modifying the cryopreservative medium, but otherwise it had been pretty well tolerated, and there was no other toxicities noted with the allogenic NK cells. So the final cohort analysis is pending, but the Kaplan-Meier curve that you see here is pretty impressive, where you can see that previous studies looking at carboplatin alone or carboplatin with doxorubicin have a survival of 15% or less. However, even with the preliminary efficacy cohort in this patient, they're already seeing a much better outcome of survival with carboplatin and the allogenic NK cells. So utilizing this preclinical data and the canine data, we looked to see whether we could translate this into our human patient. So looking at whether this was safe in human patients, expanded NK cells have been given in many infusions in over 200 patients in many different phase 1 and clinical trials, mostly against leukemias and melanoma patients. The doses were studied in sort of dose escalation studies in various different tumor types, and doses up to 1 × 10⁸ NK cells per kilo were used with no reported DLT. We also, in looking at our sarcoma population, wanted to make sure that we used chemotherapy that had known NK cell synergy. We know that NK cells can see the sarcomas mainly through the interaction of the activating receptor NKG2D and its ligand. And these stress ligands should normally not be present on healthy cells, and many chemo regimens are known to induce these ligands due to genomic stress on the malignant cell. So after much consideration, we decided on using a backbone of gemcitabine and docetaxel. So that led to the development of our study, MCC21704, which is a multi-institution study of TGF-beta imprinted ex vivo expanded universal donor NK cell infusion as adoptive immunotherapy in combination with gemcitabine and docetaxel backbone in patients with relapsed or refractory bone and soft tissue sarcomas, quite a mouthful. But the short name for that is the TINKS trial. So the primary objectives of our study are to determine the safety of this combination, because as until now, we have not used expanded NK cells or TGF-beta imprinted cells in combination with chemotherapy with this patient. So that is our primary aim. And then also to determine the 6-month progression-free survival in relapsed sarcoma patients with this combination. We have a variety of secondary and exploratory objectives, mostly to look at the toxicities, the response rates, evaluate circulating tumor DNA, and evaluate some of the other correlative studies with the synergistic effect and density, phenotype, and function of these cells, and assess overall survival. One of our exploratory objectives is to look at the utility of using iRECIST criteria for evaluation of response to cellular therapy, which has not been validated thus far in pediatric trials, and we thought this would be a good opportunity to use that over the normal RECIST criteria staging. So just some brief points about our eligibility. Patients over 12 and under 40 years of age will be eligible at this point. We'll be enrolling patients with relapsed osteosarcoma, Ewing's, rhabdo, and non-rhabdo tumors. Patients should have measurable disease per RECIST 1.1, no more than 4 prior lines of systemic therapy, no prior cellular treatments, CAR-T or expanded manufactured or cellular products. At this time, no CNS metastatic disease will be included. Due to the concern for pseudoprogression with NK cell therapy, we are excluding patients who have any tumors that are near critical structures where transient swelling could be causing substantial symptoms. There's mixed data about the effect of use of systemic corticosteroids on NK cells, but for the purpose of this study, we won't be allowing systemic corticosteroids outside of the supportive dexamethasone for chemotherapy. So what the study is going to look like is up to 8 cycles of 21-day cycle. Each cycle will be 21 days long, and patients will get gemcitabine at 675 milligram per meter squared on days 1 and 8, get docetaxel, 75 milligrams per meter squared on day 8, with dexamethasone on day 7, 8, 9 to support the potential hypersensitivity reactions from docetaxel. Due to the known immunosuppression and bone marrow suppression that can occur from this chemotherapy regimen, patients will get pegfilgrastim or biosimilar at day 9, and 72 hours later, they will get an NK cell infusion of 1 times 10 to the 8th cell per kilo intravenously. And this can be repeated up to 8 times as long as they do not have progressive disease. Disease evaluation will be done every other cycle. Local control will be allowed after cycle 2 per treating physician discretion. So just a quick overview of the study design, because it's a little bit unique compared to many other clinical trials. There will be a part 1 with 4 distinct cohorts. In the osteosarcoma cohort will include 5 patients in part 1. And as long as we can see, we identify 1 patient who achieves a 6-month progression-free survival in part 1, the similar cohort of part 2 will open up. So cohort 1, stage 1 will be opened if we see that positive result on part 1. There will be an interim analysis for efficacy after cohort 1, stage 1 is complete. And if we see at least 2 patients out of 9 have achieved our statistical parameters, then that will open up into stage 2, anticipating that each disease subtype will have at least 20 patients on this trial. I talked a little bit about the correlative studies. So these are going to be done at 3 separate labs across the country. Peripheral blood will be sent to Dr. Dean Lee's lab at Nationwide Children's, where he'll be looking at the analysis of TGF-beta imprinted NK cells in blood. And then there will be, if there are patients who have tumor resection, those tumor tissues will be shared with Dr. Mulanax's lab at Moffitt Cancer Center, and he will be analyzing the tumor microenvironment. And as we continue to build on our knowledge of circulating tumor DNA, we are collaborating with the Hayashi lab at University of Colorado to look at that at frequent time points during the study. So our current status, we have had FDA approval last fall, and I'm pleased to say that we got our final IRB approval just yesterday. So that was very exciting. Thank you. And the manufacture of the allogeneic TGF-beta imprinted NK cells is underway at our GMP facility with at least 3 batches that are ready as the last time I had a meeting. So we anticipate that we'll be open to enrollments for patients at least like late summer of this year, and hope that we can bring this novel therapy to many institutions. The study will be opening through the Sunshine Consortium, and so we anticipate that there will be at least about 20 centers across the country who will be able to enroll patients. So I'd like to thank Dean Lee and all of his work through his lab or other collaborators, the Sunshine Protocol Committee led by Dr. Isaacoff and his team, who've been really instrumental in shaping the clinical trial, our cell therapy team, regulatory and radiology team, all of our collaborators and funders, and then of course the patients and families. Thank you.
Okay, I'm still Katie Janeway, and I'm still very grateful to be here, and I want to thank the organizers for allowing me to take the closer position, not only for this excellent panel, but also for the overall meeting. So I'm here to talk to you about the Children's Oncology Group osteosarcoma trials, and this is the outline of what I'll speak about. I actually want to take the opportunity to tell you a little bit about the COG and the Bone Tumor Committee, COG short for Children's Oncology Group. I'll tell you a little bit about our priorities for osteosarcoma research, and then since this is the clinical trials session, I'll get to what our active osteosarcoma clinical trials are and our future trials that are in development. So this map shows the open or the enrolling sites for the Children's Oncology Group, which has been in existence for 20 years. There are 200 research sites throughout the United States and 27 international sites. And we think that about 90% of children diagnosed with cancer in the United States are cared for at a COG member center. And there are overall 50 therapeutic enrollments per week on clinical trials. That's all diseases, obviously not osteosarcoma. That's just showing the international sites. So the COG is organized as follows. There's an overall group chair and vice chair who sort of manage all of us who work/volunteer for the Children's Oncology Group. There are disease committees, and I'm showing you the names of the disease committees there, with the committee where the osteosarcoma clinical trials are developed being called the Bone Sarcoma Committee. Actually, we call it bone tumor. And then that's sort of where all of the disease expertise lives. But clearly, designing trials for osteosarcoma is not just a pediatric oncology effort. We need to collaborate with our pathologists, our radiologists, our radiation oncologists, and our surgeons. And so we work with these domain and discipline committees for these trials. This is a picture of the Bone Tumor Committee, at least part of our membership. It's a very large committee, and circled in red are some of the key individuals on that committee, and their names are below. So that's the introduction to the COG. Now to speak about the Bone Tumor Committee priorities. Oh boy, some formatting issues in my slides. Oh, bad ones. Okay, that's okay, not to worry. So the title of this slide says that our primary priority is to perform clinical trials in osteosarcoma and Ewing sarcoma, but for this talk really is about osteosarcoma that is focused on improving cure rates and decreasing treatment side effects. The figure that's on this slide is really meant to show what are all the things that we need to do to design those clinical trials and really conduct the best trials in this disease. We need to support basic science research. We need to identify new drugs that are active in osteosarcoma. We need to define disease subtypes, and we need to understand what the optimal approaches are for surgery. The tools or the ways in which we do that are shown on the right. Phase 1, 2, and 3 clinical trials, biobanking and specimen and data sharing, and data analyses. Okay, and I'll touch on each of these as I go through the slides. So on this slide, I want to start to talk to you about defining disease subtypes. And this is really, for those of you who talk to me often, know that this is kind of, well, I don't know, I'm a little bit like a broken record on this topic. No high-grade cancer is one disease, okay? So there is not a single high-grade cancer that's been well studied that is one disease, meaning does not have subtypes. Osteosarcoma will not be an exception. There will be disease subtypes. So I want to try to explain what I mean by that. And to do that, I'm going to talk about breast cancer. So this is a table from a recent review of breast cancer. And I think people are very familiar with breast cancer because it's quite a common cancer. And the first question that gets asked to anyone who has breast cancer is, what type is it? Okay? We don't think of breast cancer as all being the same. And this table basically shows the different types. There's hormone receptor positive breast cancer. There's HER2 positive breast cancer, which you've heard about a little bit, related to osteosarcoma. And then there's something called triple negative breast cancer. And when you look in the middle part of this table, the therapeutic approach differs for each of these types of cancer. And that's because the subtypes are defined by something underlying the cancer that kind of makes it grow, makes it tick, makes it divide, what we call a driver. So for hormone receptor positive breast cancer, that's hormone receptors. And the treatment is an anti-hormone receptor. For HER2 positive or ERBB2 positive, that's ERBB2 or HER2, and the treatment is against that HER2 positivity. So, the really important thing about defining disease subtypes is as follows. The first is that we can identify patients at diagnosis for whom MAP chemotherapy is less effective, what we call treatment resistance. That allows us to think about an approach where we give alternative treatment from the time of diagnosis instead of what we do right now, which is we observe some behavior of the cancer, what we call poor necrosis, 10 weeks into treatment. If the subtype is related to biology, that driver that I was talking about, it helps us narrow the search for new drugs for that subtype, okay? It focuses our attention. It also lets us identify patients for whom MAP chemotherapy works well, in other words, treatment responsive. And that may help us think about things like local control. You heard from Dr. Healy that there's this relationship between local relapse and how well the chemotherapy is working. And it may help us think about how we might approach our local control, for example. It also improves the interpretability of clinical trial results. That's like a statistics 102 lesson that I'm neither qualified to give nor have the time to give. Okay, you just have to take my word for that. Okay, so I said osteosarcoma is not going to be an exception. There will be disease subtypes, and this slide is meant to sort of show you some preliminary data. I don't know why the nationwide thing is on my slide. Anyway, this shows you some preliminary data. Bhuvan has taken over my slides. This shows you some preliminary data. One of them is showing how you can use the DNA or genes or instruction manual in the cell to define those disease subtypes. This is data presented at CTOS in 2019 by Dr. Lau. On the right is showing some RNA, which is sort of like the supervisor of the machinery of the cell, and how that RNA can define those tumor subtypes, some of which are more sort of immunoresponsive and some of which are less immunoresponsive and might affect how well you respond to immunotherapy, for example. And then we've heard earlier today at this conference about epigenomics and that there might be some sort of differences in the cancers defined by epigenomics. But all of this data that we have so far is what we call preliminary. So all things that are associated with treatment response and resistance, again, another statistics, probably 103 lesson, need validation. So you can find it in a patient population, but you need to prove that it's true with something called validation in another patient population or through some statistical tools, which really you can only apply in very large patient populations. And you also need to extend it beyond the original population where you observe that. And we think all of this, the Children's Oncology Group sort of hopes to play a role in doing that because we have a lot of bank tumor samples from patients who will receive sort of uniform therapy. Also, we are interested in looking at clinical features that might define disease subtypes. So, for example, we learned about lymphovascular invasion today from Dr. Blank. That would be a great opportunity for collaboration with COG. We're looking at poverty, race, and ethnicity and how that impacts how patients with osteosarcoma do with chemotherapy, similar for nutrition. And we're also interested in the extent of treatment side effects. In terms of defining disease subtypes by biology, we've looked at genetic risk for developing osteosarcoma. And we like to assist scientists who are studying osteosarcoma in the lab to assess the relevance of their findings to osteosarcoma in people. That's achieved through this biobank, which has, thousands of specimens from hundreds and hundreds of patients. And Dr. Crompton, who spoke earlier in the conference, supported by the Quad W, has streamlined access to those samples. And also AOST 2031, which I'll tell you about as one of our trials that is open for enrollment at the time of diagnosis for metastatic patients, is really going to amp up our sort of sample submission guidelines and hopefully allow us to bank really high quality metastatic samples. So now to tell you about the clinical trials. Okay, so AOST 2031 is really aimed at addressing that question about the optimal approach for surgery. This is a phase 3 randomized trial of thoracotomy versus thoracoscopy in oligometastatic osteosarcoma. The rationale for this trial is that the standard of care for resectable pulmonary metastatic disease is surgery, but approaches differ. So depending on the center or the surgeon where you're receiving treatment, you may have an open thoracotomy, which is where there's an incision, or you may have a thoracoscopic procedure where a scope is put inside the chest. Surveys suggest that there's comfort with surgery selection by randomization, meaning a flip of a coin, in those situations where either surgery would be appropriate, according to the surgeon's judgment. Because the truth is that we don't know which of those two surgeries is better in terms of controlling metastatic disease in the lungs. So our primary objective is to determine if open surgical resection is superior to thoracoscopic resection for thoracic event-free survival, meaning controlling that disease in the lungs. Patients with metastatic disease that's considered oligometastatic, which means a relatively low burden of metastatic disease that could be resected either way, who are less than 50 years old, are eligible. After patients enter, they'll have a central review of their imaging to ensure that they actually meet the criteria of the study. If they meet criteria, then their surgery will be randomized to either be an open thoracotomy approach or a thoracoscopic approach. And as I mentioned on the last slide, with the help of Dr. Jessica Davis and colleagues who you heard about earlier in this conference, we have a robust plan to bank and study metastases, and Dr. Ryan Roberts will be doing some real-time single-cell RNA sequencing of these samples, and we will also be creating a more diverse group of models working with Dr. Sweet Cordero. So we have a very important clinical trial under development, AOST 2032, and this trial is aimed at determining whether new drugs can improve cure rates and study the side effects of those new drugs. So this is what we call a feasibility and randomized phase 2-3 trial of cabozantinib in combination with MAP chemotherapy for newly diagnosed osteosarcoma. The rationale here is that cabozantinib and other drugs like it, like regorafenib, which you heard about from Dr. Davies, are active and recurrent osteosarcoma. The question that we're asking is, if you add cabozantinib to MAP chemotherapy, can we improve outcomes? So first, though, we need to determine whether it's feasible. What are the side effects of adding these two drugs together? And then once we determine that it's feasible, meaning the side effects are not too burdensome, we're going to study the addition of MAP chemotherapy, the addition of cabozantinib in a randomized fashion in both localized resectable osteosarcoma, metastatic osteosarcoma, and pelvic osteosarcoma and unresectable osteosarcoma. This is the first protocol for newly diagnosed osteosarcoma in 20 years, and it's expected to open in the winter of 2022 or early in 2023. And we will leverage the opportunity to bank tumors and study biology and quality of life. This is a schema of the trial, which I'm over on time, so I'm not going to spend a lot of time reviewing. And I just want to tell you about our active trials in relapse. So the purpose of active trials in the relapsed and refractory setting is to really focus on identifying those new drugs that might have some activity in osteosarcoma. So the first one of these is a phase 2 trial of DS-8201A in recurrent osteosarcoma. And here the rationale is that in HER2-positive breast cancer, which I told you we sometimes see HER2 expression in osteosarcoma, this drug, DS-8201A, which is also called TDXD, has basically demonstrated very good activity in a randomized phase 3 trial, and those are practice-changing results that were just presented at the ASCO annual meeting. This trial actually is sort of ongoing with enrollment, but right now is temporarily closed while we analyze those preliminary results of the first 9 patients enrolled on study. AOST 2121 is a phase 2 trial of OST 31164 after resection of recurrent pulmonary osteosarcoma. This is the same drug that you heard about from Dr. Mason's work in dogs, and that's really the rationale for conducting this trial in humans. The primary objective here is to determine whether this agent increases the 12-month disease control rate in patients with recurrent completely resected osteosarcoma, and to assess the safety and tolerability of this agent in humans. Patients are eligible if they're 12 to 39 and weigh at least 40 kilograms. They have to have lung-only recurrence and be within 8 weeks of having that lung-only recurrence resected. This is open at about a dozen institutions, and the sponsor here is OS Therapies. We also collaborate with what we call our DVL, or Developmental Therapeutics Subcommittee, to develop trials of new drugs in osteosarcoma. And we have 3 enrolling trials, which I'm showing you here, of sort of brand new drugs, and looking at the activity of those in osteosarcoma. 2 of these are monotherapies, a FACT inhibitor and tegavivint. And one of these, ADVL-2121, is studying surufatinib in combination with gemcitabine. And with that, I'd like to thank the COG Bone Tumor Committee leadership and membership, the protocol chairs and vice chairs, because we can't do this work without them, and the patients and families who have or will participate in the trials that the COG conducts and our banking studies. Thank you very much.
All right, so I think we have until 4:30. We probably went over a little bit, but we probably have about 20 minutes for questions. I see.
We're good till about 4:30.
Okay. There's a hand back in the back corner. It's John.
Thank you. This was a really great talk. I guess a general question for everyone in the group. We heard some really exciting trials for patients who have relapsed, and some of them said that you could enroll after first recurrence. And I guess just a general question, when there are active relapse regimens like ifosfamide, etoposide, regorafenib, a few others that we've used, when would you suggest considering clinical trials for your patients?
Who wants to answer that question? It's an excellent question. So I would say it's an excellent question. So I think the issue that you're getting at is kind of what is the effectiveness of what we think of as standard chemotherapy treatment for relapse. That has actually never been very well studied in osteosarcoma. And so that's why that's a very difficult question to answer. There is the trials that you heard about here are what we call prospective trials, where we enroll patients at the time of relapse, and we have some very clear indicators of whether or not we see activity. What we understand about our standard relapse regimen, which depending on who you talk to is either ifosfamide or ifosfamide with etoposide, is that we've never done a trial exactly like that. What we have from looking backwards at patients who've received it is that there may be very modest activity at best. And so because of that, I think it's very the only thing we know that you need, if you can have surgical resection, have surgical resection. And then beyond that, it's very reasonable to consider, frankly, at any time that there's relapse, first relapse or subsequent relapse, both chemotherapy and clinical trials. That's my kind of approach to this question. But there's a huge amount of variability in the clinical scenario for each patient with relapsed osteosarcoma, and so those decisions are best made on a individual patient physician kind of level, in my opinion. I don't know if other people have better answers for that.
I would completely agree, Katie. So I think for the expanding on the topic of very individual. If a patient has rapidly growing disease, then probably an immunotherapy trial at that moment is not the best choice, but it might be. And that's probably a scenario where you are thinking more, okay, let's get some ifosfamide going. But thinking about a clinical trial is always the right thing to do, to at least think about it and take it into consideration for every individual.
And I would just add that I think all of these are excellent clinical trials that you heard about, and it's really nice to see so many clinical trial options from so many different groups.
I have a general question. So it's very interesting to see, I haven't practiced medicine in 5 years, so this is a very enlightening conference. But I am struggling with, all of these are very nice initiatives, but how do you then pick what you're going to take forward when a lot of them are single-arm studies and you don't really have a good comparator? For instance, the trial with gemcitabine and docetaxel with NK cells, at the end, you'll get X% of response. How did you know how that compares to patients in those characteristics using gemcitabine docetaxel alone in order to move it forward? And I'm not picking on you. I mean, could be the same could be said about every other trial, right? How do you pick what you're going to take forward, and how do you make certain that you're making a survival advantage for patients that have a poor prognosis, not just giving them some therapy that might work?
Bhuvana, you want to try and take that, or are you, since you got picked on? I can help.
And Dr. Sethi, if you could.
I think that to a degree, I mean, when you've talked about the design of that trial in particular, you know, your patient population for each of the diseases, we've looked at what the, essentially, in the setting of measurable disease, what the 4-month event-free survival or progression-free survival is for each of the stratum. So if you're just talking about osteosarcoma, which we are, you looked at what the 4-month progression-free survival was based on, you know, the prior data from COG, and use that as a historical control, so that if you saw, so you set your statistical model so that if you saw essentially a 40% disease, or if you saw that 40% of patients did not have progression at 4 months, then you would deem that a success and want to carry that forward for further study. So there's not a randomization or a comparator prospectively, but using that historical data from the prior COG studies helps, I think, you know, in cases like this and in other studies, to set those parameters. I don't know if that completely answers what you're asking, but.
Yeah, I just want to add that in addition to
I'm sorry, Dr.
historical
Sethi,
data,
if you could pull the mic closer to you, please.
In
Thank you.
addition to historical data, we also had, I'd say, pretty conservative, like tight statistics for this study compared to many other early phase trials. More so because this is the first, in sarcoma trial to utilize imprinted TGF-beta cells in combination with chemotherapy. And so to kind of make sure that we are capturing adequate safety and make sure that we're understanding the efficacy of this combination best, the statistics are a little more tighter in a sense, that the expected progression-free survival of 6 months has to be met at those time points before we proceed to the next step of that study.
I might answer. So Nesa, Richard used to ask me this question all the time. So I've thought about it quite a bit. And the issue, the challenge with the historical COG data is we had sort of proven that those patients did not receive any effective therapy. We all think that gemcitabine docetaxel may have some activity in relapsed osteosarcoma, even though the sort of standard clinical trials said it didn't. And so the problem is that maybe it's just the gemcitabine docetaxel. I think that if we have positive phase 2 trials of combination therapy, like Dr. Sethi's trial, or the Memorial Sloan Kettering trial, the ZNC3 trial, there may need to be a follow-up randomized trial that looks at the sort of standard chemotherapy versus the chemotherapy plus the new agent. That may need to happen, just so that we're certain that we're taking, that we need the full combination and that it's not just the chemotherapy. That may need to happen. It depends. I think if the biology is really compelling, that everyone is doing in combinations with these trials, maybe we won't need that. It may be that the activity is so great that we can say that's not just the chemo. We know enough from retrospective studies that can't just be the chemo alone. I guess what I used to say to Richard all the time is this is a problem I would really like to have. I would like to see a positive combination trial in osteosarcoma, and then we can talk about what to do next.
As a follow to that, though, I guess, since I didn't give you the first ability to answer in that question for Bhuvana. There were reasons why you didn't use ifosfamide, and you did choose gemcitabine and docetaxel, which in some of those strata not only may have a low effect, but essentially have no effect. But tell us why you chose that combination versus others.
So there was definitely a lot of debate about what the right combination to use was, and as you saw in the study design, we chose to include osteosarcoma and other sarcoma types. So rather than using different backbones for different subtypes, we wanted to try to get something that was uniformly active in all of these different disease subtypes, and gemcitabine docetaxel seemed to fit that bill. In addition, we also realized that a lot of patients may have already seen first or second line relapse regimens before they enroll on study. And just from clinical practice, gemcitabine docetaxel is generally used as third or later lines of treatment. And so we anticipate that we'll be able to capture more patients on this trial that way as well. And then last but not least, of course, the biology of these chemotherapy agents in conjunction with NK cells. I think the data was very compelling in terms of stimulating the synergy between the chemotherapy and the NK cell. So hopefully with all those reasons, we chose the right combination. But regardless, I think we know that gemcitabine docetaxel has some modest activity among all of these subtypes, especially in osteosarcoma, and so we hope that we'll see synergistic benefit with the NK cells in combination.
Ryan?
A quick question for the parents in the audience. So for many of you, you're seeing parents and patients sitting here in the audience. For those that are just getting under their journey, Dr. Davis, Dr. Sethi, Dr. Janeway, and even Dr. Kaplan, and I don't mean to omit anybody else that's up there. For those that saw Dr. Davis's slide with multiple centers getting started out, but it's not the one that your child is at, how do we go about getting enrolled and getting our child enrolled in one of those programs? And not have to travel a major distance.
Exactly. So I would love to tell you that we can make it happen. But the reason that we're trying to open as many sites as we can, as much across the country as possible, is to make the travel not be 3,000 miles, but it still very well might be many hundreds of miles. We, for our particular trial, I would love to have tons of cash and be able to support people to travel. That's unfortunately not possible in many of these smaller studies. And it's also at this time not possible to give the study treatment outside of a very closely followed approved study center for a variety of reasons that include safety and regulatory reasons.
Can I add something to the response to that? When we talk, I think it's really important for us to remind people what the purpose of clinical trials are. So, and let me talk specifically about phase 1 and 2 trials, because the phase 3 trials are open at 200 different sites and hopefully near a site that your child is being treated at, if not at the site. Phase 1 trials, the goal is to determine the safety and the dosing of a medication. That means we know very, very little about that approach, okay? And a phase 2 trial is to figure out how an approach, either one treatment or 2 treatments, how well it works in osteosarcoma, which means until the trial is complete, we don't know how well the drugs work, okay? So participating in a trial is a great thing to do. It helps us learn about osteosarcoma, but it is for us to learn more as a community. I mean, all of us, not us sitting up here. And it should be, the decision about that trial should be made understanding that. It is true that we're excited. We use the words excited and promising because we get excited, but it doesn't mean that we know yet for clinical trial medication, how well it's working. I just want to make sure I make that point.
No, thank you for
not
sharing
that we don't
that.
all want you all to travel to where we're doing these trials, because we all want to learn more, but I think it's really important for us to emphasize that about phase 1 and 2 trials.
Duly noted. Thank you.
I think also it depends on the trial, in part to answer that question, because I mean Dr. Davis gave one answer, but I think Bhuvana might answer that differently for her study. Do you want to mention what? Yeah.
So I think one of the challenges that we realized when we were building this study is that for many immunotherapy trials, patients have to travel to the site of treatment. So one of the advantages of our study is that because these are off-the-shelf product, that they can be safely shipped to the treating institution. Dean Lee and his lab, they've already done and they've perfected the process of shipping with various other clinical trials. So that's not entirely new for this patient or this particular clinical trial. So, so when, eventually when we get this trial up and running at about 15 to 20 centers, I hope that patients will find this trial closer to where they are or at their home institution and not have to travel across the country for that.
And Dr. Kaplan, I believe, was trying to say something. Sorry.
Yes, I just want to say that the pediatric oncology branch at the National Cancer Institute is actively gets referrals from patients all across the country. And as being on the end of those referrals and discussing with families, I work very hard to guide families to trials that are open, that are closer to home for them, or that are more appropriate therapy options. Because even though I think we hope that every patient is treated at I do anyway, at a children's oncology group site if possible. That would be the best goal. Some patients are maybe they're young adults and they're getting access to care differently, or there are other reasons why their care might not have been, and we try very much to get them connected to a pediatric oncologist that is familiar with sarcomas, that will have very important and relevant advice for them and for their care. And so we try very hard to guide families on different available trials. The other thing about the National Cancer Institute is when we do have trials, they are sponsored and paid for by the taxpayers' dollars, and therefore not involved in insurance at all. And we pay for travel for patients that are on study. As exciting and happy as that sounds, it's still travel, and that's not really an answer for many patients that are working and living locally and then have to travel for trials. So we know that it's helpful, but it's only one part of that. And ideally, as Katie said, I think we would have a system where people would feel very comfortable going to and being on a trial, part of this children's oncology group, and or local to closer to their home than traveling far. But the trials we do are have that benefit, and it's for everyone in the country.
So we're just about out of time, but Dr. Roberts had his hand up. There might have been one other. Maybe we have time for one.
We're gonna finish it up with Dr. Roberts, and if we could try to get the answers as tight as possible, so we can get to the closing remarks, that'd be awesome. Thank you so much.
I just think it might be timely and appropriate to bring some conversations that many of us have been having kind of behind closed doors into the open. And I think we are in a transitional point in the care of osteosarcoma, and we're transitioning from a time when it was very frustrating because we didn't have very many good ideas to a time when we probably have more good ideas than we have patients. And that's going to require prioritization, and that's going to require some thought in the way that we do things, but it's probably also a moment of opportunity. And I think that it would behoove us to, whether that's through master protocols or templated protocols or just some agreed upon alignment, to make sure that we're capturing the same endpoints and that we're getting the best biology possible out of every trial, and doing some thought into how we go through and work our way through all of these good ideas is something that would probably pay off for many years into the future.
We'll keep really tight. We agree.
Agreed.
Thank you.